it will be interesting for you, actually. Indeed, so I'm presenting this in behalf of many people, of course. We've been working on this kind of on the data that I'm going to present together with Gaël Uzu and Jean Martins. Each of us having some specialty and not keeping it in our own silo. And so we try to work all together. And I will first show quite a few things that we are doing in the group, actually. The general objective of our group is the improvement of source apportionment of PM, not only biogenic PM, but generally speaking, all PM, all sources of PM, and the improvement of the evaluation of this impact on human health. For doing that, actually, we are working with offline samples and filters with a lot of different field campaigns. You have a map of the different sites where we've been doing measurements in France, for example, but also we are doing also many campaigns in collaboration with many other people in many countries across the world. And most of our samples are actually high-vol daily samples, sampled on quartz filter. And but we are doing also some work on some personal exposure samples and low volume indoor samples in answers to some of your teams. We indeed have many yearly series in order to be able to have a good view of seasonal cycles, which are really important when you are talking about different kind of sources of PM in the air. After that, the second tool we are using is indeed analytical development for tracer measurements. And of course, we are making analysis of a major minor constituent of the aerosol, ECOC, ions, trace metal, trace elements. But we are also developing quite a few other tracers or indicators for primary biogenic organic aerosol, PBOA. We are using fungal spore tracers like sugar alcohol, arabital mannitol, and we are also looking at plant debris with the cellulose measurements. We are not so many people across the world doing cellulose measurements in the aerosol, I guess, but we have a good handle on that by now in the lab. We are also doing a lot of different measurements in the lab, and but we are not applying them all the time on all series of filters, of course, because it's really costly. But we are looking at PAH, alkanes. We are doing isotopic measurements that can be interesting at some point for looking at specific sources and these kind of things. And then when we have all this database, what we are using and developing is a source apportionment with a PMF, positive matrix factorization approach, using US EPA PMF5 toolkits. And for those of you who are not familiar with that, it's only working on a matrix of observation for a number of chemical species and trying to express this as a contribution of the given sources with a chemical profile of these one sources and trying to minimize the residue. That's a reduction data process that is fairly simple on the basis, but that need a lot of improvement in order to look at, for example, reduction of the uncertainties or putting into it the proper traces in order to get the sources and these kind of things. With this, we've been working quite a bit on that for about 8 to 9 years now, and about 30, 40 papers dealing with that, with improvement of tracers, including isotopic measurements, estimation of uncertainties. We provided a database for a large EU intercomparison within the European Fair Mode initiative. And so we produced also quite a few studies looking at harmonized ensemble of PMF studies over 15 sites in France and compare that with AMS measurements, aerosol mass spectrometer measurements, and which is another type of characterization of aerosol. And we also are in the process of looking at the comparison with results of chemical transport models that have a tag source into them. The second part of our strategy is indeed to the improvement of the evaluation of the impact of PM on human health, approach with the oxidative potential of PM. So we are making development of the measurements using different assay and tests. And we are coupling this with the PM chemistry and sources in ambient air, and looking at cross studies with epidemiology and toxicology. We are in the process of introducing OP into a chemical transport model also, and we use that already in a paper in Nature published by the beginning of the year. Actually, that's 2021. And we are also in the development of a prototype of near real-time OP measurement, and we had a patent on that very recently, actually. When it comes to that, for the development of oxidative potential, we are practicing now different assay with DTT, AA, DCFH, development of glutathione measurements and direct production of OH radical also. We made a paper with Frank Kelly a few years ago already for intercomparisons. That's one of the few papers of intercomparison of measurements on OP oxidative tests. And we are in the process of starting the coordination of EU intercomparison for the OP test and the groups that are practicing that in Europe. So far in our database, we've got about 8,000 samples that has been measured for at least 2 of these assays. And you can see, for example, the results of a 7-site series in France, including over 700 samples. And you can see that there is a very good correlation with OP-DTT and monosaccharide, which are indeed tracers for biomass burning into the atmosphere. But indeed, the chemistry is interesting, but it's not telling all the story, because we clearly know that it's not, for example, the levoglucosan that is making the correlation with OP-DTT. That's probably some other things like pH, oxidative species, and kenones, and these kind of things. So in order to overcome this problem, we've been coupling the PMF chemistry and the sources with a process where you start with the chemistry. Using PMF, you get the sources, and then you try to deconvolute the oxidative potential with the sources in order to get specific sources of OP with what we call intrinsic OP per sources, that is OP per microgram of each sources, and we can get sources contribution for that. We've been playing with indeed a linear inversion for the OP, but also with neural network in order to try to take into account nonlinear processes and like a cocktail effect, actually. And so we applied that pretty recently in a paper that is out now with on a 16 yearly series of samples in France. And what we got is indeed this kind of results where we can have an average mass representation of the average of these 15 sites in France. You've got it in microgram or in percentage. And compared to that, you can have the mean OP with a DTT or the mean OP with the AA assay. And indeed, for 8 different sources, the sources including sulfate-rich, road traffic, nitrate-rich, and dust, HT salt or biomass burning, and also primary biogenic traced by actually the polyols I was mentioning earlier. And what you can see in that is the representation that you get from for the PM repartition between the source is really different if you look at it with a mass or if you look at it with a different OP test. And for some of the sources that are important in the mass, as nitrate rich, they are not at all important for the OP measurements in the series. And for the biomass burning, which is already a pretty important species in terms of mass, is all the more important when you look at the different OP tests, for example, the OPAA. And if you look at also at the road traffic fractions, the road traffic fraction in terms of OPA results, it's much more important than it is for the mass. So you can rank the different sources according to that with the 15 sites that we have in the study. And you can see that when you are considering mean mass, the first important source in terms of mass is biomass burning over France, and primary biogenic are only 1 to 6 sevenths in terms of importance. But when you look at the OPDTT or OPAA, the primary biogenic is coming up in the list and getting something like a 5th rank into this ranking of the importance of sources. This is indeed because when you look at the intrinsic OP of the different sources, here they are ranked with a distribution of what you can get across the different sites, and with also like a Monte Carlo approach of the uncertainties that you have on this intrinsic OP, you can see that the primary biogenic indeed has quite an important intrinsic OP compared to other sources that were ranked before in terms of mass. And it's nearly the same, at least for the OPDTT, the intrinsic OP of primary biogenic is pretty much like the one from biomass burning. It's not the same thing, of course, for the two measurements of the OP. Intrinsic OP of primary biogenic for AA test, it's much lower, actually. So this pushed us to work a little bit more on the primary biogenic sources, and we had a PhD thesis on that. That was kind of pretty productive, actually. And we've been able to look at time series of these different sugar alcohols tracing fungal spores into a large array of sites over France. And first of all, we can see, of course, and this is well known, that these species are much more in the PM10 fractions than in the PM2.5 fractions on the sites where we had comparative samples for the two size fractions. Then you have a good grip on what is a seasonal cycle for this species. Here you've got many different sites across France, going from rural area, urban in alpine valleys, or traffic sites in urban areas. And you've got a concentration for average concentration for seasonal variations. And you can see that maximum concentration are in summer or in fall. And it's not necessarily actually the rural environments that have the highest concentration. Some of them are indeed in urban alpine valleys, and we still don't know exactly why, but most probably because the alpine valley are not subjected to long-range transport from outside environments, and mostly the air in the summertime is much more impacted by local variability. And what is interesting is indeed is that with a large array of sites, we've been able to look at the time series of concentration and look at the correlation when you look at the distances between the sites. And what you can see is indeed for sites that are within something like a 150 kilometer range, you've got very similar variation, time series variation, meaning that at least for this primary biogenic linked with fungal spore, the sources are regional sources on the range of about 100 kilometer, 120 kilometer, which we hypothesis in the paper is linked to the dimension of similar biomes and homogeneous biomes, which are and climatic biomes which are producing fungal spore in the same way and with similar processes. With this study, what we've been able to check is what is a representation of the organic fraction that this PBOA is producing. And you can see that on a yearly average, on the left side of this figure, the PBOA bore an amount about 10–20% of the total amount of the organic matter of the PM. But when you go into the summer average, you can have something which is above, on average, 25–30%, and it can go higher. Again, particularly in the alpine valleys, which are isolated environments, and they are prone really to a large fraction of this PBOA being constituting the OM fraction of the PM. And when you look at the profile of the PBOA that you obtain with the PMF results, you can see that the polyols are making up only 3% of the total organic fraction, meaning that there is a lot of organics which is not characterized in this process, but and you've got association with a few chemical species associated with that. Most of the profile is indeed organic matter. When we've been looking at a little bit more closely on a very rural site in eastern France, indeed that's a rural site where there will be disposal of nuclear waste and in the deep soil, and we are maintaining an observatory of there with long-term measurements. When we are looking at that, you can see that indeed the polyols that we are looking, their concentration is increasing a lot during the time of harvesting. For the other different agricultural practice, the link is way less than that. And when you are looking at the same time at cellulose measurements, you can see that the cellulose have a very good correlation with the polyols, and the cellulose fragment can reach very high concentration because it's above 1 to 2 microgram per cubic meter, which is extremely high compared to what you can see. Well, it represents a very large fraction of the organic matter. So this was really interesting, and indeed this led us to look at what is the links between the microbiology with fungi and bacteria in these samples, trying to compare what we can see in aerosols in the leaves of the vegetation that is around the site and in the soil. And because a few of the papers that are dealing with fungi and their emission process, they make the hypothesis that the emissions are coming from the soil. And definitely in this case, we can see that it's not at all the case. The association, both for fungi and bacteria, it's much larger when you are looking aerosols and leaves than what you are when you are looking at aerosols and soils. So definitely in this case of a very rural area with large agricultural fields, the association is much more, and the emission process is much more with whatever is related with a vegetation than with soil remobilization. And this opens somehow the way of making parametrization in order to put that in chemical transport model, because so far there are really few studies looking at the process of emission of this kind of species into the atmosphere. And by the end, what we've been looking at is indeed trying to link specific, measurements of specific fungi and bacteria species and the DTT test. So what we've been doing is making some cultures into monocultures of bacteria and fungi into the lab and trying to do tests on this type of material. And what we get is indeed, first of all, testing that with a pure biological material. Oh, sorry. And what we see is that there is all tested bioaerosol, both fungal spore or bacteria, give a good, a good signal with OP-DTT. And increasing with the load of the microorganisms. And actually the fungal spore OP-DTT is much higher than the one from the bacterial cells. And then, I'm sorry, I missed a couple of slides. Where did they go? Going to find some other ones.
Just 2 minutes, Jean-Luc.
Yeah, just the matter to find them again. Yeah, can you see that now? No. Okay. Yeah, okay. Then after testing the microorganisms by themselves, we try to look at a mixture of bioaerosol and chemical species. First, we started with fungal spores. And what we can see is actually that when you are testing the mixture of copper and some microorganisms, you can see that there are additive answers for the DTT test for both copper, naphthoquinone, or the mix of naphthoquinone and copper. But indeed, when you are looking at bacteria, you've got something that is fairly different. You've got a decreasing signal of the OP-DTT for all the models, chemical species that we are testing, and not the same behavior for the different type of bacteria. And so there are several hypotheses going to that, why we see a decreasing signal, but we can talk about that later. Okay, as for the conclusion on bioaerosols, all of them have an intrinsic OP, which means that when you are testing atmospheric PM, you will find a signal coming from the bioaerosols themselves. They add up to modulate the OP of chemical species with either way an additive signal or a depressing signal. And we also tested the process with inactivated fungi, for example, after intense gamma ray irradiation. And indeed, we found out that the OP is mostly related to a passive mechanism, because it was, even when the cell were dead, it was taking place anyway. Okay. Well, that's about it. And well, here are some partners and some labs we are collaborating with and some sponsors. And I'm really thanking you for your listening this talk.
Jean-Luc, thank you very much. That was a very exciting and interesting talk.